Hey students, here is lesson number 3 from Unit 6. That was originally supposed to happen in our Zoom call today, but I'm going to record it here. There are two major objectives for this lesson. The first one is to describe how different types of mutations can impact the result of translation. And then secondly, I'm going to talk about one major exception to this central dogma of biology that is commonly referenced on the AP exam, but it's just also helpful to know going forward. Okay, so let's start with that first objective. I want to talk about mutations. So quick refresher, right? A mutation is just a change in the DNA sequence, that is the specific base pairs of DNA. Now, mutations typically occur when DNA is being replicated. So during DNA replication, which we know from Unit 4, typically occurs during the S phase of interphase, right? When we think about the cell cycle. So the S phase preceding mitosis or meiosis is where this DNA replication occurs. And as those leading and lagging strands are being copied, there can be a variety of errors that can occur as DNA is being synthesized. Now, there are a couple of different types of mutations. Actually, there's lots of different types, but I'm going to go through some of the major types that we need to be familiar with and the impacts that a mutation can have. Because if you remember, right, DNA is connected to proteins through the processes of transcription and translation. And so if a change occurs to the DNA, there are a variety of impacts, including no impact, to the protein that's encoded by the information in the DNA. So let's take a look at kind of this first major category. That first major category is actually, we're going to use this whole section here, is under the category of a substitution mutation. A substitution mutation. And all a substitution mutation is where one base or one nucleotide is swapped out for another, substituted for another. So where there was a G, now it is substituted with, for example, an A. And so that substitution, which can occur during DNA replication, can have effects on the protein. And there are kind of three different ways that substitution mutation can affect the protein. So I'm going to use these three different parts of this figure. But first, a quick overview of how to interpret this. So we're going to first talk about a silent substitution mutation. And for each of these, you're given a wild type DNA, which is just the non-mutated DNA sequence. And then you have a mutated sequence. And what you'll notice is that they've only included one strand from the DNA. We know DNA has two strands. They just included this top strand. And then they have included the mRNA and the amino acids that correspond to this DNA sequence. Now, quick note, right? Remember that DNA has two strands. So if we were to draw the other strand, right, we'd expect to see a T here, a C, a G, a C, an A, and so on and so forth. The mRNA was transcribed using the bottom strand that's not shown of the DNA sequence. And the reason they didn't include it is because since we know the DNA strands are complementary and that mRNA follows very similar base pairing rules except for there being uracils, that actually this strand of DNA will be the same as the mRNA, except instead of a T, there will be a U in that place. So that's just how you interpret the DNA and the mRNA. So we've got DNA transcribed into mRNA and then translated into amino acids. It looks like they give you 4 amino acids, 1, 2, 3, 4, with the corresponding codons. Again, that's determined by that genetic code table that's shared by all living organisms. Okay, so the substitution mutation that's occurred is right here, right? So you'll notice that on this top strand, there used to be an A, and it's been substituted instead for a T. And the impact that that's going to have is on the mRNA, where there used to be an A, now there is a U. And so in that second codon specifically, now you have GUU where there used to be GUA. Now, the reason we call this a silent substitution mutation is because we have substituted a nucleotide in the DNA sequence, but it's had no effect on the protein sequence. And that's because, if you remember, right, that genetic code table, for a lot of the amino acids, there are multiple codons that lead to the same amino acid. And so just because you change the DNA doesn't necessarily mean you're going to change the protein. Okay, that's a silent substitution mutation. A missense, on the other hand, is a substitution mutation that changes one amino acid in the amino acid sequence. And so let's look here. So we've got our non-mutated again and our mutated. And this time, the mutation that's occurred is right here, where there used to be a C. Now there is an A. And now this is going to impact the third codon, where there used to be a CCC. Now there is an ACC. And that's actually going to code for a different amino acid that used to be proline, but now is threonine. Right? So that is called a missense substitution mutation, where that substitution in the DNA changes an amino acid. Okay, and then lastly, a nonsense substitution mutation is actually where that mutation doesn't change an amino acid into a different amino acid, it actually changes it into a stop codon. And this can often cause translation to stop abruptly, right? Because this stop codon has now been created as a result of this substitution mutation. So in this case, the substitution occurs in this last nucleotide, where there used to be a C, now there is a G. That affects the last codon. That used to be UAC and led to tyrosine, but now is UAG, and that actually stops translation. So that's called a nonsense substitution mutation. So those are some possible impacts so far, right? It can be silent, missense, or nonsense, all right, depending on how or where the substitution mutation occurs. Okay, now another type of mutation, or two types of mutations, are insertions and deletions. Insertions and deletions. And instead of, in the case of a substitution, one nucleotide replacing another, just like the name suggests, an insertion or deletion is where one or two or multiple nucleotides are either added or deleted from the DNA sequence. And this can cause a whole bunch of issues. And so we're going to walk through that right here. So insertions or deletions, looking at this figure right here, can cause something called a frameshift mutation. And what that means is that if you take a look at the non-mutated or wild-type DNA right here, a deletion mutation has occurred where these two bases have been removed. And here is now the new sequence. So that T and A have been removed. And what that does, you'll notice that not only does it change that codon, right, that used to be GUA, but now is GCC, but every codon after that also gets affected because the whole sequence shifts up. All right, so now those groups of 3 called codons, that whole reading frame, those blocks of 3 have been shifted. So we call it a frame shift, and that can cause multiple amino acid changes, and this can also be pretty severe in terms of the impact on the amino acid sequence. So a frame shift caused by insertion or deletion can have large impacts on that amino acid sequence. Okay, so we've got substitutions, which can be silent, missense, or nonsense, and then frame shift, which can cause a whole bunch of changes to the amino acid sequence because it shifts the reading frame. Okay, the last thing I want to talk about here briefly is, or actually, let me go back for a sec. Real quick reference to Unit 1, right? Remember that the reason this matters of changing the amino acids is because we know that different amino acids can have different properties, right? We know some amino acids are hydrophilic or phobic or charged or uncharged, and that can affect the tertiary structure especially, because all of these amino acids can have different R groups, and that's typically what gives them their unique properties. So changing one amino acid might have a slight impact on protein structure, although there are definitely examples of a single amino acid change causing a huge disruption to the protein function. But when you consider a frame shift caused by an insertion and deletion, you have all of these different amino acids with different properties that are going to drastically impact how the protein folds. Okay. So I want to talk about this last question here. Are all mutations inherited by the next generation? So remember, right, DNA mutations occur whenever replication is happening. And so that can happen before mitosis or before meiosis. And when we strictly talk about, let's say, humans, right, we have mutations happening in our cells every day as a result of mitotically dividing cells in our body. However, only mutations that occur prior to meiotic division will be inherited by the next generation, right? Because if we think about this, mutations acquired in your skin cells, for example, that divide in response to, say, when you get a cut, right? If you acquire a mutation in the DNA of a skin cell, that's not going to be passed on to the next generation. Only DNA mutations that occur prior to meiotic division, right, will ultimately end up in the gametes that will ultimately be passed on to the next generation, right? So just a quick note, remember that not all mutations are inherited by the next generation. Okay, so we've talked about different types of mutations, some of their impacts. Let's talk about our second topic here. Okay, so the second topic that I want to discuss is a very common topic that's brought up on the AP exam, but it's often just discussed as kind of a contradiction of sorts to the central dogma of biology. And if you remember, when we say that term central dogma, what I'm referring to is the main flow of information in an organism that's described by DNA being transcribed into RNA being translated into protein, right? That's kind of the major flow that's seen across all living organisms of how genes are expressed and how we understand these common processes that occur across all of life. But it's been observed that a particular virus, which is, it's debated, but it's not an organism in itself, since it has to hijack the processes of a living cell. But the way in which it infects a cell actually shows this flow of information that is a little bit different than what we'd expect. And so let me give you just a quick overview of this virus, and then we're going to talk about why in particular this virus is said to contradict the central dogma of biology. Okay, so HIV, human immunodeficiency virus, is a particular type of virus called a retrovirus. And this virus has a couple of different components. It has something called a capsid, which is kind of this interior portion. It's a bunch of proteins that create this kind of capsid structure. And then it has an envelope. And an envelope is mainly composed of a lipid membrane. And similar to a cell, there are these different envelope proteins, such as GP120 or GP41, that are found on the envelope of this virus. And right, you've heard of COVID-19, right? A lot of the talk is about spike protein. That's an example of an envelope protein that's specific to the SARS-CoV-2 virus that causes COVID-19. But in particular, HIV retrovirus that has these specific envelope proteins, and inside of its capsid, this interior structure, there's a couple of different things. The main thing I'm going to talk about right now is that it has a particular type of genetic information. It has single-stranded RNA that contains the information that's needed to replicate the virus. Now, there are lots of different types of viruses that contain different types of genetic information. Some have DNA, some have double-stranded RNA like COVID-19, but this particular virus has single-stranded RNA. And currently what we know about HIV, and for many of these viruses, including COVID-19, there is something called a zoonotic spillover, which just describes where this virus was naturally circulating in other animal species and crossed over or spilled over into humans. And this particular virus had been observed to be closely related to the simian immunodeficiency virus that was circulating amongst African primates, and due to random mutation and contact with humans, this virus was actually spread or spilled over to the human race. Other things to note that this virus specifically targets what are known as CD4 positive T cells. That's a particular type of immune cell in the human body, and that has to do with the type of envelope proteins that this virus has. So you might have heard, going back to COVID-19, right, that those spike proteins are specifically good at targeting cells in our lungs. That's why it's a respiratory illness. But this one particularly affects the immune system, which is why it causes AIDS, right, which is the condition known as autoimmunodeficiency syndrome, where your T cell count drops beneath a certain level because they're being attacked by HIV, and so you are extremely susceptible to infections such as influenza or pneumonia, which often is the result of, or which often results in death of someone with AIDS, is that secondary infection of another illness like influenza or pneumonia. Okay, so if we actually take a closer look at the RNA, we can kind of get an idea of what's encoded by the instructions in that RNA. And so this is a figure that actually represents the genome of HIV, that single-stranded RNA. Now, there is a lot of stuff that we could talk about this, but what you'll notice are these different sections that correspond to producing particular proteins that are needed to replicate the virus or make more virions. And similar to, what we see in the human genome, right? The human genome has all of these genes that code for particular proteins that do specific things. And so the HIV genome is about 9,700 base pairs in length. And across that 9,700 base pair genome, there are these genes such as GAG, POL, and ENV. And these different sections correspond to, for example, the GAG region produces or stores the information needed to produce proteins for making that capsid, that structure in the interior of the virus, and other core structural elements of the virion, the new HIV virions. The POL region encodes a particular enzyme called reverse transcriptase and integrase, and some other enzymes called RNase that are needed both for supplying new virions, but also in the infection process. And then the ENV region, these are the genes that are responsible for making those envelope proteins like GP120 that we talked about in that previous slide, that are kind of on the structure, on the outside surface of the virus, right? So those are kind of the three major elements of the genome that store information to make those proteins. So how exactly does HIV infect a cell? Well, so let's take a look at this model. So this model right here kind of gives us an idea of what HIV does. And so HIV, it will use that GP120, so starting up here, it'll use that GP120 envelope protein to anchor to these CD4 receptors on these T cells. So this is a T cell inside the human body. It's a type of immune cell, CD4 positive. And that interaction actually allows HIV to anchor and ultimately fuse its envelope, which again is a lipid membrane, with the host cell, the T cell's lipid membrane. Kind of like endocytosis in a way. Back to Unit 2. And that releases that capsid, which contains a whole bunch of things that the cell or that the virus needs to replicate itself. So we have that viral RNA. You'll see it contains already reverse transcriptase packaged in that capsid, ready to go, as well as integrase and a couple of other proteins that the virus needs. And what's amazing is, remember that that genetic information is actually RNA. And here is where it's said that HIV contradicts the central dogma. Because the central dogma would predict that as that RNA gets inserted into the cell, it should be translated, right? RNA to protein, it should be translated. But instead, reverse transcriptase, like its name suggests, actually takes that mRNA and reverse transcribes it into DNA. And with the help of some other proteins and a pretty amazing process, it can essentially convert that viral RNA into double-stranded DNA as a result of that reverse transcriptase working. Then, integrase, like its name suggests, is an enzyme that will actually take that viral DNA and will integrate it into the host genome. And this is why HIV in particular is very difficult to treat and cure, because that viral genome actually gets integrated into the host genome and is treated like any other gene in the human genome. And so then this integrated viral DNA can then be transcribed into RNA and translated into the proteins that that RNA encodes and can then be packaged with some of that RNA into new virions. Right? So we see the packaging of the RNA and those proteins. We see the appearance of those envelope proteins as we progress this way. And then ultimately, that virion is released. Right? So the reason why it's said HIV kind of contradicts essential dogma is because we start with RNA and we go to DNA, which then can be used to go to RNA and protein. So there's kind of a reversal of the direction of essential dogma initially, before then it flows through DNA to RNA protein to make new virions. So I've also included, for comparison, how SARS-CoV or SARS-CoV-2 functions, so the virus that causes COVID. And it actually turns out that SARS-CoV-2 is a is an RNA virus as well. I think it's specifically a double-stranded RNA. But you'll notice that, right, it interacts similar to what we see with HIV, where there's this envelope protein called the spike protein that interacts with these ACE2 receptors that are commonly found on respiratory cells. And it allows the virus to interact, anchor, and fuse with the cell. And as that occurs, that RNA is released, but you'll notice that that RNA is immediately translated, right? Unlike HIV, where that RNA was reverse transcribed, integrated, and then transcribed and translated. This SARS-CoV-2 or SARS-CoV immediately has the RNA translated. And then through the help of some other enzymes, RNA copies are made of the viral genome, and the proteins that are made by translation are all used to make new virions, right? But slightly different in terms of the flow of information. This follows kind of the central dogma, where this with HIV tends to contradict it. Okay, so I hope that was helpful in explaining why HIV in particular contradicts essential dogma. I'll also include a really cool animation for you to view about the HIV infection cycle to kind of get a clearer idea of this reverse transcriptase enzyme and integrase as well. So reach out if you have any questions, but have a good rest of your day.